Good morning, everyone. Now, moving towards our second session, it is my honor to introduce our second speaker, Dr. Subarna Roy. He is currently working as scientist and deputy director at ICMR National Institute of Epidemiology, Chennai. He has completed BSc in botany from Hooghly Mohsin College, University of Burdwan, MSc Botany in Microbiology from University of Burdwan, and PhD in Microbial Genetics from ICMR National Institute of Cholera and Enteric Disease, Calcutta, University of Burdwan. He has served at ICMR institutes in the field of microbiology and molecular epidemiology of infectious diseases. He has worked in state administration and district health surveillance program for identification of infectious disease outbreaks, for monitoring of drug resistance patterns, and for prevention control. He has undertaken several molecular genetic studies on authentication of medicinal plants, documentation and preservation of germplasms, and genetic fingerprinting of medicinal plants of Western Ghats. He has also led the Biomedical Informatics Center of NITM, linking medical research with information technology. He has more than 100 peer-reviewed articles with citation 1370 and H-index 20 to his credit. He has been awarded with Major General Saib Singh Sokhi Award 2009 from ICMR in 2013, Dr. S.S. Misra Memorial Award from National Academy of Medical Sciences in 2008, and Medical Innovation Fund Award from Indian Council of Medical Research in 2006. So we are here very fortunate to have molecular epidemiologist Dr. Subarna Roy for this FDP program to share his valuable thoughts on herbal informatics in drug development. Over to you, sir.
Thank you. Thank you very much, madam.
Yeah, thank you so much, sir. You can start sharing, sir, now.
Fine. Are the slides visible?
Yes, sir.
Yeah, fine. So, thank you, ma'am, for a wonderful introduction. And it is my pleasure to actually speak to you in this faculty development program. And I am basically a biologist, so I'm not an informatician. So it will give you a good mix of hardcore artificial intelligence that you had in the previous session, followed by a biological perspective. And the very purpose of this particular faculty development program and the topic that has been chosen is very apt, because this is the fringe area where the informatics and the biology merge together. I never liked mathematics in school, and therefore chose biological sciences as a career. Maybe many of the faculty here also in my group. But then now things so confusing, very difficult. There is a lot of scope and opportunity in those areas where the subjects merge. And this herbal informatics, as I call it, is one such area. And as Madam has said, that I was working with the National Institute of Traditional Medicine in Belgaum for the last 11 years. And then I have shifted to National Institute of Epidemiology. So I had the opportunity to work with a lot of pharmacists, a lot of medical personnel, and then try to find out what we can do actually to merge the different upcoming areas together so that we can find solutions to present health issues. And as it happened, uh, in traditional medicines, we generally work with not only the areas of Ayush, but also in areas where the folklore medicine is concerned. The traditional practitioners, whom sometimes we call as quacks, and the community service providers, particularly in villages and in tribal areas, and
Hello, sir.
some of them, yeah. Can you?
I'm sorry. Yeah, I'm sorry to disturb you, sir.
Yeah,
But I
please.
think there is some disturbance from your side. Uh, can you use the headphone or something sort of that? This
Uh,
probably will remove
is
the
there some noise?
interference of sound. Yeah, there is a noise in the background.
Okay, maybe because of the fan, I will switch it off.
Yeah, yes, sir.
Is the noise there right now?
No, no, it's fine now, sir.
Yeah, I think it was the fan which was creating the
Yeah,
problem.
yeah, I think so. Yeah, thank you
Yeah,
so much, sir.
yeah. Thing is that I am not in my office today, so I'm yet to join back. I
Yeah,
have,
we had,
yeah. So I had come home, so I don't have the gadgets with me, so I'm a little bit limited with the infrastructure.
no,
Yeah,
no problems, no problems.
yeah. So that's what now what we are going to do. Perhaps this will be very important in your respective areas where you are teaching and doing your research. And basically, we are looking at the traditional medicines which are used in folk medicine or in Ayurveda or in Siddha or in Unani, and then trying to find modern remedies for some of the diseases that are actually plaguing us. And what more occasion than the present outbreak? Because as you all know, there is no drug, there is no vaccine, and everybody is trying whatever is possible by repurposing various drugs which are available, and even repurposing of traditional medicines. So that is why the importance of informatics and the data boom in research and drug development is extremely important. I will make things easier when we go through the contents one by one. And the first part is just basic introduction about the traditional medicines or herbal medicines and the classical drug discovery pathway, followed by the bioinformatics as a tool that we use in our drug discovery process, computer-aided drug designing and bioinformatics, strategies that we generally adopt to discover drugs more efficiently using these bioinformatic tools, and the commonly used databases and softwares that are available at your disposal to use. The next gen informatics and the big data biology that we are talking about today, and the future perspective. So these will be the nutshell of what I am going to talk to today. I will give one example with one of the work that we have done. So herbal medicine or botanical medicine or traditional medicine, whatever you call them, are based mainly on herbs and plants. And they are practiced over centuries. So they are practiced through folk culture, maybe even through organized systems like the Ayurveda in India, the Chinese traditional medicine in China, the Siddha, Unani, even homeopathy to an extent. So these are practiced long time over history. And you all know that a major portion of the modern day drug have actually originated from natural sources. Even artemisinin and also even Nobel Prizes are being awarded for that. So there has been a renewed thrust in traditional and folklore medicine, because of certain reasons. One is the high price of modern day healthcare, adverse events that are mostly associated with modern drugs. More importantly is the lack of penetration into the masses, particularly in the rural areas, where still access to modern medicine is very poor, mostly in developing and underdeveloped countries. These are your slides in pharmacy, so you know much better than me. This is quite an old slide where actually it has been projected. The herbal drug industry has a potential to grow immensely over the next few decades, and it has got over 3.2 metric tons estimate of the trade of raw drugs of botanicals, and more than 960 medicinal plant taxa are in trade in India, and a priority list of 176 plants have been made by the NMPB, which are in trade. And again, this is your slide on the classical drug discovery pathway, where we have known targets but unknown pharmacokinetic suitability, safety, efficacy, and we have a compound library from which we design the drugs, which are more of the target-based drug design. Then we have what we call as leads, and then we go into preclinical research, followed by clinical research, and then we come to the drugs, which is a very long process, and it takes almost 15 years for a drug to be developed, and from thousands of drugs, you will get only one approved drug. So the pipeline is a very, very narrow sieve that it goes through, and its costs are huge. So to actually overcome these bottlenecks, several strategies are adopted. The first strategy is to reduce cost, and to do that, simulations are done, the computer-based simulations of the activities of the drugs. To increase data, we generally multiplex to several things together. High throughput, so equipmentations through high throughput, high output, bulk outputs, we generally increase the data in short time. To save time, we parallelize. Sometimes we do many things together at the same time so that we save time, and time is money. To minimize human errors, we generally automate, so we don't have much of human interface, and then reduce the chances of human errors in the drug discovery process. So therefore, one must be able first to generate the data and to cope with this large amount of data that is generated in a short span of time. So in a very short span, what we do is simulate, multiplex, high throughput, parallelize, automate. So we generate such large data. So we need to cope up with them. And most importantly, we must have trust on the process that has generated the data. If we don't have trust in that, if that process is not correct, then you are not going to get good data. So that is extremely important. So what are the bioinformatic tools that are available for simulation? First, I said about simulation. So here we have various areas opening up even today. We have already some which were used for quite some time, but new disciplines, new avenues are opening up in this area every day. Here you use them for identification of target disease. You can use them for screening of interesting compounds that you may get. You can use them for detection of the molecular basis of the disease in question. You can use this for rationalization of your drug designing techniques. You can use this for refinement of the compounds that you have already got on efficacy and or adverse events. You can use them for quantitation of the structure-activity relationships. You know them very well, QSAR studies. And determination of the physical properties like solubility of molecules, testing of the drugs, and similar. I will come to them individually, so that will make things much easier. The first is the identification of target disease and interesting compounds. So first, say you want to identify a disease that you want to target. This year, everybody is targeting COVID-19. So anything that works on COVID-19 or apparently works on COVID-19 is being tested. So you need to identify which disease you are going to test or you are going to find a remedy or a cure for. Now then you have to find is that all that is known about the disease. You have to identify the key and the key metabolic players that is determined. Again, I think every month you are hearing something new, that it is affecting the kidney, it is affecting the gut, it is affecting the brain, it is affecting your renal functions. So other than the earlier known pulmonary involvement, there are so many other involvements, other organs as well. So these are kind of informations that are coming through research, and those things are taken into consideration while you are designing a remedy or a suitable drug to actually address this COVID-19. Then what you can do, or what is being done, particularly in the case of COVID, is that known existing molecules or those from hypothesized remedies are identified and their structures are determined. Remdesivir is the classic example. It was designed for AIDS and for a completely different disease, but then it has been repurposed. It is just not that just we have something and then we try it. There has been a lot of bioinformatics studies like this behind, which actually likely shows that it may have some effect on the SARS-CoV-2, and therefore this is being tried. So that is how the first thing to do is that whatever are available molecules, those are tested against these particular disease targets. It is also important to look at very similar afflictions of their known treatments. For example, say a lot of studies are going on those particular drugs that are used in the common cold or flu. So if they have some kind of a cross protection, so those kind of studies are also going on. So those are also very important to understand and take into consideration. But you must understand that identification of the target is not sufficient to achieve a successful treatment of the disease, because these are all theoretical and a real drug actually needs to be developed ultimately. The bioinformatic methods like the docking are used to virtually screen the targets for compounds that bind and inhibit a particular protein. In silico docking studies are carried to find out what we call as the lock and key fits. So and a ligand which bind together like as in a lock and key. The more perfect the fit, the better it is as a drug candidate. Now you must understand there might be multiple locks and keys involved. So a key may be having multiple locks, or a single lock and multiple keys. So those kind of situations also arise. This drug must influence the target protein in such a way that it does not interfere with the patient. So naturally, for any drug development, it should target the disease in question and not the normal metabolism. Otherwise, you will have adverse events or side effects. At most times, the compounds that are likely to have some activity against these diseases are generally screened. And they may be sometimes in the very beginning only marginally useful and may have some side effects also. Then what happens is that these compounds provide a starting point, and then they are refined chemically or in their structure in artificial manner or in the laboratory, or some derivatives are made of that, so that the side effects are and their activity is increased. So then you have to determine the molecular basis for the disease. So if it is known that a drug binds to a particular spot on a particular protein or a nucleotide, then this drug can be tailor-made to bind at that particular site. This is often done or modeled computationally using computers with a variety of techniques and softwares. The primary way of determining what compound would be tested computationally is generally determined by two approaches. First is the researcher's understanding of the molecular interactions, or by brute force. So here I want to mean if which are the compounds that we are going to test against what. This, who determines that and how is it determined? So this can be determined in two ways. First is that you have a thorough knowledge about the disease and the compounds that are involved in this disease, and you also have a thorough knowledge about the possible drug targets that you are using. And then you use your own brain to understand or to test out which are the compounds that you will be testing against these disease targets. So this is the researcher's understanding. On the contrary, there may be another strategy where you use brute force method. So that means whatever compounds are there which you want to target, and whatever compounds you want to use as drugs, you test them all and then see whichever comes out more suitable. So if you have a high throughput system, if you have a high resources, then you can use this brute force method for screening of your likely drug candidates. Then comes the refining part of the compounds. Once a number of potential lead compounds have been found, then computational laboratory techniques are used in refining these molecular structures to give them better activity and less of side effects. This is done both in the wet lab as well as in silico studies using computers by examining the molecular structures to determine which aspects are responsible for both the drug activity and the side effects. So once you identify the domain of that structure which is responsible for the drug activity, you modify them to make them better. And if you identify the domain which is involved for the side effects, you try to remove them. Drugs are then modified structurally to achieve the desired effects, and you may be knowing the CADD, the computer-aided drug designing, is a specialized discipline that actually uses these computational models or methods to simulate these drug receptor reactions. And CAD methods are heavily dependent on these bioinformatics tools that we talk about and the applications and databases. As such, there is a considerable overlap between CAD research and bioinformatics. And there is now even further overlap with, because now artificial intelligence is used in a very, very elaborate manner into most of the research applications. And here also a lot of artificial intelligence is used nowadays for drug discovery. So pharmaceutical companies are always searching for new leads to develop new drugs and compounds. One such method, one of them, is the VHTS, the virtual high throughput screening. In this, the protein targets are screened against databases of small molecules or large molecules, compounds, to find out which of them binds strongly to the target. If there is a hit with a particular compound, it can be extracted from the database for further testing. And with today's computational resources, several million compounds can be screened in only a few days of time in sufficiently large or clustered computers. Pursuing handful of promising leads for further development can save researchers a lot of time and expense. As an example, zinc is a good example of VHTS compound library. So zinc is a database which can be used for this kind of screening. In CAD research, one often knows the genetic sequence of multiple organisms or the amino acid sequences of proteins from several species. It is useful to determine how similar or how dissimilar organisms are based on their gene or protein sequences. With this information, one can infer even the evolutionary relationship between organisms, search for similar sequences in bioinformatic databases, and find related species to those under investigation. This is what has happened with the SARS-CoV, the original one, which caused the original SARS earlier. So a lot of drugs which have been tested against SARS are being used in SARS-CoV-2. So because of this, because there are similarities in their sequences. There are many bioinformatic sequence analysis tools that can be used to determine the level of sequence similarity. Homology modeling is an important challenge in CAD research, and it is the determination of the 3D structures of these proteins, because proteins, as you know, they are not planar, they are in three-dimensional forms, and it is that three-dimensional structure only which acts as the lock and key. So therefore, the determination of the correct 3D structure is extremely important. Most drug targets are proteins, and therefore their knowledge of their 3D structure is also of importance, so that you know both the lock and the key kind of structures. And it is estimated that the human body itself 5 lakh to 10 lakh proteins. So you can understand to what extent these 3D structures need to be known. However, only a small fraction among these proteins we know the 3D structure. The remaining are modeled, or the computer actually gives out the possible or predicted structure, 3D structure of these particular proteins. So in homology modeling, the amino acid sequences of specific protein targets is unknown, and the 3D structures of the proteins related to this target or the template are known. The bioinformatics softwares are then used to predict the 3D structures of the target based on the known 3D structures of templates. For example, when the 3CL Pro and spike glycoprotein structures of the SARS-CoV-2 sequence were obtained, their 3D structures were not actually done through crystallography initially. They were modeled based on similar structures found elsewhere in other viruses. And then later on, the actual structure was published. So initial, in January, February, the drug developments were based on these model structures. Later on, the actual structure came, which is more or less similar to what was modeled, and then the quest for drugs actually became more streamlined. So modular is one very well-known tool in homology modeling, and so is Swiss model, a repository database of these structures created with homology modeling. So this is a screenshot of the modular software, which can be used to actually derive the 3D structures of the sequences of proteins that you may actually submit to this this resource, and it will generate the models. And once the models are generated, it can be actually stored in this Swiss model, and you can download these 3D structures from these online resources. There are other softwares like Schrödinger. It is a very commonly used software. However, this is a paid software. And here you can do a lot of these docking studies, the simulations, the 3D structure determinations. Autodock is mostly used for docking the target and ligand binding happens in this Autodock. SwissDock is also another example of such softwares. Now, another approach is the subtractive genomics approach. This is often used to find new putative drug targets which aid in drug discovery. So you are identifying the drug targets in a particular disease. The proteome consisting of all the known proteins of a pathogen, if it is an infectious disease, is retrieved from the database. So say we have a pathogen A and we know all the protein sequences so far known about this particular pathogen A, and we retrieve this proteome from the database, and then paralogs or duplicates that are present, several proteins may be present in duplicates in this particular organism A. So we remove the duplicates with the help of, again, online resource like CD hit web server at a certain percentage threshold, which results in a subset of proteins. So we bring it down to a lesser number of proteins that we want to target. Then we can use BLAST P, which is generally performed against human proteome. So now we are comparing this list of proteins which are present in this particular pathogen A against those which are present in human to identify non-homologous protein sequences. So there may be some sequences of proteins which is common both in the pathogen as well as the human. So that we subtract. So to identify only those proteins which are present in the pathogen A but not present in the human host. Then the proteins are subjected to DEG BLAST P, which is called the database of essential genes and genomes. So then whatever remaining we have the proteins of the pathogens, we need to see if they're essential for the pathogen. If they're not essential for the pathogen, if you even target them, then the pathogen will escape. If it has to be a drug target, then it has to be essential for the pathogen. So from this database, you identify which of these proteins are essential for the pathogen. Then what you do is you find the subcellular location of these proteins, which can be predicted using softwares. So where each of these proteins are present in this pathogen, whether it is in the cell membrane or whether it is whether it is in the cytoplasm, so whether it is in the nuclear material. So those kind of things you will come to know using you can predict using the softwares. Then docking is done with your hypothesized remedies or your proteins of interest which you think may be the known drug, maybe your possible drug candidates. This is similar to the reverse pharmacology approach. You may be knowing about the reverse pharmacology approach of Dr. Vaidya. And here what is done is that we take those remedies which are already in practice in traditional medicine, be it Ayurveda, in Chinese traditional medicine, in Siddha, Unani, or even in folklore medicine. Say we have some cuts or some bruise, we use some haldi powder or something to just disinfect that area. So these are known remedies, and the safety and efficacy is also known. So we know that these are safe, but we don't know how it happens and what is the target. So it is the opposite of the pharmacology approach. And here we have clinical evidence, then we do preclinical research and try to understand the ADME, the adverse events, if any, and then perhaps identify even single molecules from them or even the fractions in the phytopharmaceutical pathway towards drug development or establishing the efficacy and safety of these known traditional medicines. So that is the reverse pharmacology approach. So we have databases of several medicinal plants that are related to our kind of research. and numerous of them are available, and you can just go to these websites and you can try to find out and get this clinically used or traditionally used medicinal plants against various kinds of diseases. The Natural Medicines Comprehensive Database, the NMCD, HerbMed, National Library of Ayurvedic Medicine, TKDL, the TCM database, Medline Plus, Supernatural 2, Herb Ingredients, Target Hit, then the database of medicinal materials and chemical compounds of Northeast Asian traditional medicine. These are all databases. You also have PharmDBK, which is an integrated biopharmacological network database for traditional Korean medicine. So they have, these all databases have quite some overlap in between, but then many of them have something different, and some of them are, say, the AfroDBs for African medicinal plants. So the TCMs are mostly for the Chinese medicinal plants. The TKDL is of the Indian origin. So the National Library of Ayurvedic Medicine for the mostly plants used in Ayurveda. Again, there are some common plants which are used in across the systems of medicine. So there will be some overlap, but each of them offers something new or something different than the other. Then you have databases of bioactives. So you have the bioactive compounds. The UNPD is the classical example for this. So and you have Dr. Duke's phytochemical and ethnobotan. Here you have the phytocompounds listed in these medicinal plants. So all the details about these bioactive compounds you get from this online resources. And this is how things are generated and then put into the database. So you have your wet lab studies, you identify the compounds once it is established, and then they are put into the database. So that is how the database grows. And for chemistry, again, UNPD is one good resource to have this. The ChemBL is a very useful online resource for the chemistry of medicinal plants. Drug Bank is also one of them, which is very often used. For targets of disease, you have the binding DB, the binding database, where actually a lot of disease targets are listed, and you can get the sequences and the 3D structures of the disease targets from. Even the chemical database also provides some targets. SwissDoc, I talked about earlier when you do docking studies, so they also will provide some information. Protein database, PDB, this is a very important resource. You get a lot of information about the proteins that you are looking for from this database. UniProt is also one which is a very commonly used database. Now, once we have disease target associations both together, then the TTD, the therapeutic targets database, is one which can be. You have both the disease and the target association results put in. The Human Protein Atlas is also trying to make such kind of interactions known in the human, and it is also growing in a very fast pace. And then there are other databases like disease and gene annotations. Then network development and system biology analysis. Now, so far as I was talking was more of a particular disease, a particular target, and a particular solution or a particular drug candidate to be used for that particular target. But what happens in practice actually, a systems approach happens where you have multiple metabolic reactions occurring in the body. You have one drug which has multiple reactions and multiple effects elsewhere also, not only in the target, but most likely it can affect some other aspects, some other reactions also. So that is how things happen in practice. And this is what is being stressed in the last 10 years on what you call as systems biology or network pharmacology. And you are not concerned only about one particular point where actually it hits, but also its repercussions in the entire equilibrium of the body system that happens. And with these resources coming in, like you have networks which can be altered by the addition of a particular drug candidate in the system. So those kind of databases are also coming up in a big way and are more likely to help answer questions about adverse events or side effects or even complementary effects, particularly as far as traditional medicines are concerned. David is also a bioinformatics resource software which actually gives some information about the system's biology. String is also very frequently used software for this kind of purposes. Now, this is a screenshot of the Chinese traditional medicine database at Taiwan. And you put in your username, password, and then you get a list of the traditional medicine database, news, updates, basic search, advanced search. You have a lot of services that are offered by this particular database. It is one of the largest and non-commercial database for TCM, and it has a collection of information from Chinese medicinal texts and scientific publications together. So it constitutes more than 350 different herbs, animal products, and minerals, contain about 37,000 non-duplicate TCM compounds. It also provides comprehensive ingredient structural information about each of these traditional medicine. And original research articles also of these ingredients are available in the reference section. And you can have the molecular file formats provided in the database that you can actually download and use for docking studies and molecular dynamic simulations. So you can search this database as per your requirement through the chemical composition or through the plant or even through their properties or through their structure. This is another traditional Chinese medicine integrated database. It has got more than 8,000 herbs listed there with more than 25,000 ingredients and 6,800 drugs, and has 17,000 targets and more than 46,000 prescriptions across 3,700 diseases. So this is also a quite comprehensive database that is available on Chinese traditional medicine. The herbal ingredients target database, the HIT database that I talked about earlier also. This is how the screenshot of its homepage looks. And it currently contains about 1,300 known protein targets. 221 are proteins are described as the direct targets. 200 literatures which cover about 586 active compounds from more than 1,300 reputed Chinese herbs. And it contains detailed interaction values such as IC50 and KDKI. They are collected, and it's possible to get these values also from this database. The UNPD comprises about 2.5 lakh molecules, and it allows template-based search for the molecule or the natural source of these molecules. Supernatural 2 again is a very well used by. It contains a database of natural products, both small compounds and synthesized by living organisms, and contains around 3.5 lakh natural compounds with their 2D structures, not 3D. Then physicochemical properties, their predicted toxicity class, and the potential vendors from whom you can actually get these compounds. The chemical diversity of these molecules are tremendous and often offers inspiration for innovations in medicine, nutrition, agrochemical research, and life science. Most of the currently used cosmetics and drugs are either natural products or close derivatives of these particular products. It provides information about the pathways that are associated with synthesis and degradation of natural products, their mechanism of action with respect to structurally similar drugs, their target proteins, It allows a template-based search for similar compounds or come with the compound names, even vendors, and the specific physical properties or their substructures. There are other various databases which are linked to some specific disciplines. This Nube database is more of biodiversity database, sustainable resources. So it contains natural products and derivatives from Brazilian biodiversity. Contains main chemical and biological properties, the 3D structures and compounds. It's a useful tool for studies on naturally occurring bioactive compounds, their molecular and physical chemical properties, database generation, virtual screening, and deduplication, metabolomics, and other study design for drug development, mostly with Brazilian medicinal plants. Natural Product Alert is also another database which is available to you for very similar kind of work. And it contains around 2 lakh scientific papers and reviews, mostly on natural products. And mostly their ethnomedicinal information, their pharmacological and biochemical information, whichever are available, including those studies which are done in vitro, in situ, in vivo, and in humans, as well as in clinical trials. So these are further details about the contents of this particular website. Detail, but there are several categories of diseases and the activities that you will find in this particular website. TKDL is India's own resource. If you can remember, a lot of issues happened with patenting of basmati and haldi, and that is the time when India realized the importance of publishing whatever India's traditional knowledge exists. And this was created with that objective quite some time ago, and so that people cannot, outside people cannot patent them. And these are for common use in Indian medicinal plants. So this is again Indian resource. This is Ayush Soft, mostly on Ayurveda, and it is created by Ayurveda physicians, mostly based at Pune and Bangalore. And this gives a lot of information about those treatments that are available in traditional medicine, particularly for Ayurveda. Now I will share with you just a couple of slides of one example of how these resources are used for your routine studies. In one of the studies that one of our students had actually tried, he was trying to find out the alternative remedies for diabetes mellitus, which is a very common disease affecting a good number of people in our country. And it seems there are quite a number of traditional diabetes, including the BGR therapy. So they contain quite a number of listed plants which are used in management of diabetes mellitus. And by looking at literature and the traditional practices, we shortlisted 11 plants. We thought that we will test these 11 plants using only bioinformatic tools, whether they can be useful in treatment or management of diabetes mellitus. So what we did is that we mined all the known phytocompounds from this particular 11 plants. Now these 11 plants are used by various folklore practitioners all across Western Ghats in India, and many of them are actually reported even in modern literature to be useful, but nothing much is known about their molecular mechanism of action. Neither there is any established paper or review done on their efficacies and their safety. So what we did is that we identified these 11 plants and then listed out all the known phytocompounds that are present in them and which have been submitted in any of these databases that I talked about. and then downloaded their structures. And many of them had their 3D structure available. Some of them did not have their 3D structure. So those 3D structures were modeled in computer. And then quite a number of proteins were obtained out of that, including their structures and properties. And then what we did is that we tried to understand how many number of the chemical reactions that are involved in the biology of or the disease progression of diabetes mellitus. Around 26 chemical reactions or metabolic reactions we found out, and then we tried to use this all this compounds from these traditional medicines from these 11 plants, and then try to dock them together, whichever works best for particular metabolites in these 26 reactions leading to diabetes mellitus. And then we ultimately screened them, and then we shortlisted 63 phytocompounds which were predicted to target 26 proteins which are associated with diabetes mellitus. So our network was drawn of them. You can see in the bottom of the slide is that network was drawn, and these 63 phytocompounds were predicted theoretically using computer simulations to target 26 proteins associated with diabetes mellitus. Then again, 82 enriched molecular pathways were identified. And from them, through the KEGG pathway database, we identified 21 targets that modulated 11 pathways. So it was even further shortened using the approved the models which are available in the web resources. And then further compound target pathways were derived for each individual as well as in a network pharmacology or polypharmacology approach. And then the phytocompounds that acted on multiple targets were also determined. Say, for example, quercetin, epigenin, and several other compounds were found to target multiple areas in these reactions, in various reactions. And then we determined the adequate properties of drug-like ligands. Some of them passed and some of them failed. So whichever has failed, we removed them, and whichever passed, and those were taken for the biological spectrum of the selected phytocompounds. So that is how we have actually reduced the dependence on laboratory studies. We have reduced time, and almost with nothing, means it does not require much of your expenditure in actually doing these studies. And most of these studies were done using freely available softwares and the freely available databases. And we published this also recently. This is going to be published, I think, this month. It has already been accepted. And so several compounds like epigenin, then quercetin, and some other phytocompounds were modeled and were found to block certain metabolites in the diabetes mellitus pathway, particularly PL3K, AKT pathway, the signaling pathway. Those are pathways that have been found to be blocked. So now, these are things, these are ways in which you can zero in on 2 or 3 compounds with a particular disease target and then take them forward in your wet lab studies. I am not saying that this is enough, but this actually reduces your dependence on your resources of laboratory, your time, but you use this as for screening and then likely mode of action. So once these are taken into your wet lab studies, and if you get activities, if you don't get much of side effects, then you can be very well assured and say that this is the reactions that actually happen in the body, and so you will have a molecular mechanistic mode of action also described for the action of this. So now, as Dr. Joshi was also talking about that there is an information boom, and we are presently living in a big data environment. data is being generated at tremendous pace. The resources, the web servers that I was talking about, if I have made these slides maybe 3 years back, if you look at the existing number of compounds now, perhaps it will be logarithmic scale, it has been increased. So that is the kind of increase that is happening in this data. And as you have seen in this artificial intelligence lectures, the more you train, the more data you actually audit and approve, the better the AI becomes. The same thing happens here. The more the data, the better the reliability. If you remember, I had talked about that you should have complete trust in the system. So the more the data is generated, the more reliable they become, the more trust you have in the system, and the more likely they will be drug-like candidates. So that is how things happen. And this is the growth chart of biological databases over the years. And if you can see that there is a tremendous increase in the number of databases that are available over the years. And even in published genomes, if you can look with the advent of next-gen sequencing, you have lot number of sequences and of both the nucleic acids and the proteins coming up, and the database, the number of databases available also has taken a very steep incline in the recent years. And data is also collected faster and in greater volumes. If you can see the genomes here, the submissions, if you can see, has much outnumbered the reference clusters. So reference clusters were less, and now submissions have become so high that they have taken a very steep rise. So these are even Medline indexed articles published per year. So you have a lot of publications coming up recently. Earlier, even our PhD guides and all, they never perhaps reached even 100 publications. Because at that time, everything used to be manual, and you send papers for publication. They used to come back after review, so it takes around 2 to 3 years to publish. But nowadays, things are online. You do everything fast, and then you get a good number of publications and in quick time. So therefore, the number of indexed articles published per year also has taken a big leap forward. Open access papers are increasing over the years, so you have a lot of data available to you without a cost. So that is also taking a very steep curve. And this is a Elsevier slide. Again, the open access and then the gold open access, which is a paid kind of a thing between them. So that if you look at, so both have increased. This is actually opposite kind of a curve. Actually, it is on the right hand side you have the older, and the left hand side is the recent. So here it is the percentage of what is being written is available online. So earlier we never used to get research papers published 20 years back or 15 years back, but now you get them. So that is how things have become more available online over the years. If you look at discipline-wise publication, if you can see, these are the discipline-wise publications in open access. And if you can see the top few, first are multidisciplinary, dentistry, veterinary, pharmacology, toxicology, pharmaceutics, biochemistry, genetics, molecular biology, medicine, agriculture and biological sciences, health professions. Then comes physics, astronomy, neuroscience, immunology, environmental, mathematics, social sciences. So I want to mean is that the biologists are actually contributing most in the in this information boom. So these are a number of publications across disciplines in biology, proteomics, bioinformatics, genomics, computational proteomics. The red one, which is the top one, is the bioinformatics. So a lot of studies are being done recently in bioinformatics. In the last 20 years, it has been actually left the other disciplines far behind and has climbed very high. So therefore, the future belongs to this information sciences. And presently, the doubling time of the whole genome sequence data is around 6 months. So today, whatever information we have about the whole genome sequences doubles in 6 months. the doubling time of the data storage capability at the same cost is around 18 months. Say today you buy a 8 GB pen drive for 500 rupees. At the same cost, you will get a 16 GB pen drive 18 months later. So the cost remaining the same, the data storage capacity is doubling at 18 months, whereas the biological data is doubling at 6 months. So the cost, the output of data actually is increasing at a much faster pace than the storage capacity of the data. So you will find that storage is going to become more costly than actually the cost of generating the data in the years to come. Those of you who are using Gmail very frequently will now know that your Gmail, your 10 MB or something space that they give is going to get over. So they ask money for increasing your storage. So that is how things are happening. So the cost of storing them will be much more. Another thing which is very important is that now we are living in a world of data. So it is very important and challenging for scientists to make sense of this enormous data that we have. You will find a lot of papers coming out for COVID using the data which is available in public domain by various universities and medical institutions across the world. So that is because the data is available. So with India having centuries old tradition in herbal medicines like Ayurveda and one of the largest biodiversities in the world and providing the best of IT professionals in the world, it is extremely unfortunate that there is no comprehensive integrated digital archive of scientifically validated Indian traditional medicines and Indian databases as such. Other than TKDL, IUSoft, we don't have it in that scale as the Chinese TCM has, and that we feel should change over the next few years. And we have to take this collective responsibility for not only creating these databases, but also putting this data together, which is our own data. Before I end, I will just leave you with a small clip of how this data is influencing us. If you can, here in 60 seconds, 204 million emails are sent, 300,000 logins are made, and 1.3 million YouTube videos, 1 lakh tweets, in 20 hours of video, 62 hours of music downloads, in 60 seconds. And that is how this data has now become big data. And we are talking about petabytes and exabytes now. And the variety of this unstructured data is tremendous. And the velocity of data is real time. Big data is the next wave of business transformation, science, and it means big opportunity for all of us. And the time to act is now. So with this, I would like to end my talk and would like to take questions if any.
Thank you so much, sir, for this wonderful session. Now I request all the participants to raise the questions if they want to discuss anything or ask anything to sir. You can raise your hands, or also you can put your queries on the WhatsApp group. One question has been asked by one participant, sir. I'll raise that question by Mr. Pratap. He's asking that, is there any database for Indian herbs and medicines for vast number of diseases, as we have many indigenous herbs?
Yeah, that is a very, very important question that you said, and that is what I talked about. It is very unfortunate that we have things in very patchy manner. We do have databases, including the ICMR NITM has got a database of about 500 medicinal plants used in various kinds of ailments, and they have been curated and collected from the traditional practitioners in Western Ghats. So that is available in public domain and it is free of charge. Similarly, there are various other databases which are available in various universities. You have in various research bodies, but nothing as we can take pride in India's consolidated database. That kind of thing is not there. So, and we need to integrate all of them together into something very large kind of a database because we have tremendous data.
Yeah,
And
right.
every day we are adding to it. Yeah.
Absolutely, sir. Absolutely. Yes, one more participant wants to ask a question. Rajnikant. Sir, you can raise your question.
Sir, good morning, sir. How are you, sir?
I'm fine. Thank you. Thank you. Tell me.
Thank you for your wonderful presentation, sir. Really, you have enlightened the audience with a good number of databases on the herbals, on the natural products, sir. Thank you for the information. Sir, in your presentation, you mentioned a point on supernatural 2, sir. Sir, could you please, if you don't mind, could you share that slide, sir? I will note down that website. Supernatural 2.
Yeah,
Sir.
yeah.
Please,
yeah. Just a minute.
And is it something different from the other databases, sir? Like you mentioned about HIT database and you mentioned about the UNPD also.
Yeah, most of the databases are, each database differs from the other in some way or the other.
Yes, sir.
So what I would advise you to is look at all the databases that you can get. Then whatever you are interested in,
This one, yes.
You can actually you can actually look at them and then try to download in as per your need. And what I do is that I am also from the government side, so we have tremendous crunch of funds sometimes. So what I do is that I always use this freely available softwares and sometimes these trial versions. And then with that, I try to do whatever is possible. So my team is just like you and anybody else. So we have a shortage of resources, and we try to make the best use of whatever is available freely.
That's great, sir. That's great, sir. Yeah. Thank you, sir. And one more small query, sir.
Yeah.
So from this database, we can download the moieties also, sir. Like
you can download moieties. You can download moieties.
Download the moieties, and we can utilize them for the computational studies.
Exactly.
Thank you, sir.
Yeah, like I am a biologist. I do not know much about the technical aspects of these things. So what I do is I have a student who is a bioinformatician. So we are supposed to understand what are the avenues available, say even for artificial intelligence. I will not be able to program or whatever, perhaps you will also not be able to do it, but at least we will know these are the ways in which we can apply this into our own research, and then we will approach those people who know how to do it. So we will give them the problem and we will give them the solution, and then take the collaboration of these people, and then together we do it. So it becomes useful for everybody. So that is how
that becomes a great collaborative work too, sir. Yeah.
Exactly.
Thank you, sir. Yeah. Thank you for your valuable time, sir.
Yeah, exactly. I think one who is a domain expert and the one who can prepare a database by making up some kind of softwares or utilizing the codings in different ways, I think this will help us to search in a much better way.
Right.
Yeah. There is one more participant coming in, sir, with a question. Harshida Patel.
Yes,
You can
sir.
ask your question.
Yes, ma'am. Thank you, ma'am. Thank you, sir, for the very good session and informative session. Sir, I wanted
Thank
to
you.
know about that which database we can use, or which database is based in which we can just enter the plant name and get the constituents.
Uh, I think supernatural 2 also will give you, and it depends on what plant you are actually talking about. Uh, Say for example, those plants which are listed in the NITM Med Plants database, which belongs to NITM Belgaum,
Okay.
there we
Okay,
have
sir.
listed out the known phytoconstituents which are available. It has been listed out.
Okay.
So,
Okay, sir.
so it will more depend on the web, the database that you are using. Some people will list them out, whoever has got that plant listed. And if you
Okay,
don't
sir.
find it in some other database, you go to another one where they have listed out. So the
Okay,
ones
sir.
that I mentioned are the most commonly used databases. So you will find most of the common plants in any of them, any of the databases.
Okay. Okay, sir. Okay. Thank you, sir. Thank you, sir.
Yeah.
Are there any more participants who want to raise any question or ask anything? Okay, sir. I think there are no more questions from the participant side.
Right.
So, yeah,
So thank you, madam.
yeah.
And
Just a minute, sir.
yeah, yeah.
Thank you, sir, for sharing the information about various databases like traditional Chinese medicine integrated database, herbal integrated targeted database, supernatural 2, and so on databases, along with the details and how they are, along with the examples. We are grateful to have you and we appreciate your efforts to make this session really interesting. Thank you, sir.
Thank you, and I would like
Yeah.
to place on record my thanks for the organizers and giving me this opportunity to speak to you. Thank
No,
you.
we are thankful to you, sir. In spite of
Thank you.
so many difficulties, you managed to share your knowledge and talk with us. So we are
Thank you.
obliged. We are obliged. Sir, I think there is one more participant who has raised
Yeah,
question.
yeah.
So can I ask?
Sure, sure,
Yeah.
sure, sure, sure.
Yeah. Deepak Painuli. You can ask your question, sir. I think the participant is not
Hello.
connected. Ah, yes. Yes,
Hello.
sir. Yes, sir, you can
Am I
ask.
audible?
Yes, sir.
Yes, sir. Good afternoon, sir. It was a nice presentation, first of all. And my question is that I am a software engineer and I want to apply some my AI techniques and ML techniques in pharmacy or diagnosis of diseases. But the problem is there that I am unable to get meaningful data sets for the machine learning purposes and AI technique using AI techniques. So is there any collaborative research where the pharmacy people or medical people and software people like me can collaborate in the research? Is there any available, sir?
Yes, actually, uh, uh, there are a lot of scope there. And what you will require, as you said, is perhaps you require data set, because for any development of software, you will require a data set to be used. Then after that, you can build upon that. So yes, there are institutes which actually are doing a lot of software development and custom made apps for taking care of various issues. Uh, but what I have seen is that they are mostly doing it in house through their own, either their own project mode, where they advertise and then they recruit software engineers or some technical experts in this, and then give them the problem along with the data set. So because naturally sometimes most of these data sets will be a kind of
private
in-house kind of a thing. So that's why something which perhaps will be in public domain and then you would like to develop software will be difficult to think at the moment and find out. But then most of the institutes, research institutes, who have data not only in this drug development but in other diseases as well. The AMRs, the hospitals, I tell you, in hospitals, if I were the CEO of a hospital, first thing that I would like to have is an app where I have a continuous monitoring of the antibiotic sensitivity of all the pathogens that I am having isolated from my various patients. Because that determines my antibiotic policy, which are the antibiotics that I am going to use. So each hospital should be having that kind of a system, but they don't have. But these are things which are actually practically needed. So you need to approach those kind of institutes who are keen to have this kind of applications in mind. Best thing is that if you can collaborate with a biologist or a health professional and discuss these kind of issues and then approach together that you don't have to do anything, you just share the data and we will develop the which you can have this kind of application. So that is how you can go about.
Okay, thank you.
Yeah, thank you so much for your kind gesture, sir.
Thank you.
Yeah, thank you so much, sir.
Thank you. Thank you. Thank you.
Thanks a lot. Dear participants, although your session 3 starts at 2 o'clock, but due to some technical issues at the resource person's end,